number of these, that number would suggest that they're the majority of all biological entities. There are more bacteriophages than all other forms of life added together. There have been independent estimates for the number of bacteria, and those numbers come to about 10 to the power 30. So that makes us think that these numbers are not too far off. It's also estimated from experimental work that there's something like, globally, about 10 to the power 25 bacteriophage infections per second globally. And this would suggest that the entire phage population likely turns over in a really short period of time, maybe a few days. And so there's a lot of these things. We could imagine that they've been evolving for a very long time. And it's an extremely dynamic population. There's also been quite a lot of work done in thinking about how bacteriophages influence the ecology of the planet more globally, and the role that they play, for example, in the biogeochemical cycle. It's estimated that this abundance of bacteriophage infections may account for something like 10% of the entire carbon turnover in the biosphere, simply because of this constant infection and killing of their bacterial hosts in the ocean. So I'm just going to conclude briefly now, and I'll take a couple of questions if people have questions, and then we'll move along. So just to summarize, I've told you that what bacteriophages are. They're viruses that infect bacterial hosts. I've told you that they're numerous, that they can be lytic, but they can also be temperate. Perhaps most are temperate rather than lytic, but that's an arguable point. We've discussed the important roles that they play in the ecology of the biosphere, and indeed, this population is large but extremely dynamic as well. So I'm going to go on in the next part to talk about phage genomes and genome evolution, but we can pause if anybody wants to run to the bathroom, I guess, or we can take questions on what we've talked about so far. I can get a drink of water. Questions? There's a microphone for you.
I've just got a question about transcriptional repressors.
Yes.
Are they host-derived or phage-derived?
It's a very good question. The question is whether the repressors that shut down phage gene expression in a lysogen, those transcriptional repressors are encoded by the bacteriophage. If one is analyzing a bacteriophage genome, sometimes it is possible to identify those genes as encoding transcriptional repressors, but that's not always the case, simply because there's a lot of genetic diversity amongst that particular group of repressor proteins. They are typically expressed by the bacteriophage themselves. There has incidentally been quite a lot of interest as to whether some of those repressors may not only, they may be encoded by the prophage and regulate prophage genes, but they may also have the capacity to regulate other bacterial genes that are present in that lysogen. And that's actually a really interesting area of pursuit.
What about the mechanism of homoimmunity? How is that at the molecular level regulated?
The best way of thinking about it is that these repressor proteins, I told you that the prophage expresses the repressor, and then it binds to target sites on the prophage DNA. Homoimmune phages typically have similar repressors, and they certainly have similar binding sites or operator sites. So if two phages are homoimmune, then the repressor protein that is being made by that lysogen is able to shut down gene expression of the other phage by recognition of its operator sites in the DNA. Does that make sense? So typically, prophages that are heteroimmune have different repressors and different operator sites. So repressor specificity is the clue to understanding immunity.
Does that mean that a homoimmune cell will be infected and become a dilysogen? It'll just stay lysogenic?
There are examples. So it's a good question. So just to emphasize again, the question is, if a lysogen becomes infected by a homoimmune phage, what happens to that phage DNA? Does it become also integrated into that genome, essentially as a dilysogen, or a lysogen that carries now more than one prophage? And there are examples where that's true, and there's examples where it's probably not true. For example, there are some lysogens for which it's been experimentally shown that a superinfecting phage, even though it's homoimmune, still injects its DNA. And you can do that by making recombinant bacteriophages that carry reporter genes like GFP or luciferase. You simply infect that lysogen, and you can show that the DNA goes in and the genes are expressed, even though it doesn't go through an infectious cycle. And in those cases, the formation of double lysogens or dilysogens probably can occur. On the other hand, there are examples where bacteriophages may encode other functions that specifically modify the outside of the cell wall, and preclude other bacteriophages from even injecting their DNA. So there again, there's general concepts, and there's deviations from that. Some do it,